I think this. We're recording now.
Okay, I'll let people in. Thank you.
Okay. Yeah.
Are you doing the introduction, Katie?
Yeah, I can do it.
Okay. People haven't seen you for a while. They have seen you a few times now.
Yeah, I have been back after some time. So
that's how it goes.
So welcome everyone. I won't take time because I'm looking forward to hear Colin Sheppard today. I think Colin needs no introduction, and you can read his extensive biography on our homepage, and I won't go through it. So some questions I asked Colin just before this meeting is, is there any country that he didn't visit? And his answer was Antarctica. So you might guess like he has worked almost.
There's plenty of places I haven't been.
No, but as you will see from his bio at our RMS website, he has been to almost most major universities and places, and then like I was thinking like if there is any microscopy method that Colin hasn't contributed to, and I was thinking maybe electron microscopy. I haven't read his papers, but then Colin told me his PhD was on electron microscopy. So probably there isn't a method that you will come across which doesn't, if you trace back the papers, you will find a Colin Sheppard paper just describing the theory or experimental design of that whole method. So he has been one of inspirations of me, and actually I was lucky to edit. My first paper as an editor was Colin Sheppard's paper in Philosophical Transactions of Royal Society, which I'm fairly proud of. So Colin, I want to listen to you, and I will cut short now, but please read his bio, it's very extensive. So Colin, I'm all ears. Yeah.
Right, okay. Yeah, thanks very much.
Oh, can I just hold on a minute? Sorry, I'm having problems. There's about 20 people in the waiting room and I'm trying to let them in and I can't work out how to do it. Just 1 second.
I will try to do that. All. So I think all of them are joining.
There, got it. Sorry about that.
So my great introduction went to waste.
Yes.
Nonetheless, we will hear Colin now. Yeah.
Okay, right. So I can start?
Yes.
Good. Yeah, okay. So today I'm going to talk about my understanding of the development of various techniques that have become important in recent years. And a lot of this stuff, actually, I started getting together way back when I first went to Oxford. I went to Oxford as a postdoc in 1974. And started working on laser scanning microscope at that time, and did a lot of the first thing we did really was to do a literature search to find out what had gone on before. So I'm going to go right back, quite beginning of last century. Hold on, why? I'm not moving. There we go. Yeah, so I'm going to start with the ultra microscope, which was built in 1903 by Siedentopf and Zsigmondy. And what I wanted to point out here is that basically, the instrument is exactly the same as a light sheet microscope or even the theta microscope that Ernst Stelzer developed before the light sheet. But you see that the basic principle of this is that you illuminate horizontally, as in this picture, and then you look at the image at right angles to it. So the paths of the illumination and the detection are completely separated. So this is a neat effect, which of course, if you're looking at very small objects, the scattered light is very weak. And so you don't want to have to worry about the illuminating light getting into the imaging system. So this technique of using this offset illumination scheme is going to be something that keeps coming up time and time again as we go through, actually. Yeah, so this is the next thing I wanted to mention. You see this one also, this is by Goldman. a bit of a jump now. We've gone to 1940. I've missed out in that space the first people who did fluorescence microscopy came on the scene, and actually I think Curler, the one who invented the Curler illumination system, was one of the first people to do fluorescence microscopy around 1911, I think it was. Anyway, we're jumping now to Goldman. And you see, this is another system where you've got this offset illumination system. In this case, it's not 90 degrees, it's 45 degrees. So here's his source of light. Here's his object, which is in fact, in his case, is an eye. So this is a system for looking at the cornea. You know, this system has got a lot of interesting features that appear in many modern designs. Effectively, it's the first, I think, it's the first confocal system, or at least it's what we might call now semi-confocal, because instead of using a pinhole, it uses a slit. So here I've labeled the slit. So basically what we do is we illuminate the object with a line of light, or actually a plane of light. So it's effectively a light sheet microscope. But only this one line of the object is imaged onto this slit. So the slit acts like a confocal slit. But unlike a confocal microscope where you have to scan in X and Y, in this case, you don't have to scan in the Y direction. You produce a direct image along the length of the slit. He also came up with a really nice way of recording the image. This is a long time ago, of course, so no computers. He did it by writing the image directly onto a film. And this film was rotated as it scans. Now, this system actually, it scanned in depth. So this source was moved up and down. As the source moved up and down to focus in different depths, so this film rotates, and then you produce an image like this one here. So this is a cross-sectional image through the eye. You can see the cornea here and the lens of the eye, really quite remarkable, really, because you know, I think if you show this to modern day people, they would think it was an OCT image, not something that was done in 1940. If you look in his paper, there's a lot more detail than this. This is just one picture. Actually, most of the paper is about medical things, about the eye. So it's quite interesting that he took it so far way back then. This is actually a recurrent theme, is that there's been a lot of microscope techniques that have been developed in the ophthalmology area. I think they've been particularly inventive over the years. Okay, now we're going to jump to 1943 and Koana. So now we're in Japan, and he described both reflection and transmission systems which had confocal pinholes. These systems were built to do microphotometry. So basically, his idea was that by using a confocal pinhole, you can get rid of the stray light. So in any microscope, you're going to have some flare light that's scattered from inside the system and so on, even within the object itself. His idea was that by using this pinhole, you could get rid of the stray light and get a more accurate measurement of the strength of the light. So the other interesting thing, this was 1943. Why he was working on this in 1943, just at the end of the war, is not clear. But anyway, that was the case. So I'm going to all the slides I give are in roughly chronological order. So I'm going to be jumping around between different areas. I hope that's not too confusing, but that's the way I've arranged it this time. So now I'm going to talk about what's called the flying spot microscope, which was the first form really of scanning optical microscope. And the way it was done in the early days was by using a flying spot system. So a flying spot tube, as you can see here, is basically a television display, and the electron beam is scanned, and it produces a scanned spot of light, which we use to scan over the sample, and then we pick up an image. So it's much the same system that was actually used in the original Logie Baird television system, where in those days, in Logie Baird's day, the newsreader would be sitting there in a dark room, and he would have a light beam that would illuminate him point by point, and that was how the picture was propagated. This I came across in a book, Photoelectricity and Its Application. It's a book by Zworykin and Ramberg, which is actually a lot of that is about electron microscopy and a lot of different things. But 1949, and he mentions for the flying spot microscope someone called R.C. Webb, who I'd never heard of until I found this. Here there's a bit of a description about these flying spot scanners and using them as a microscope. Now another one from the same book, Zworykin and Ramberg. Basically, this is describes a confocal slit system, and the way they did it here was to use a lamp which has got a straight filament which acts as a line source, and then you have a detector with a slit based in front of it. So the idea there is that again you can get rid of stray light. He mentions here about microphotometers, and you see, so just like the Koana system, he's using the slit to get rid of the stray light. 1950. This is a bit of a different subject, but this is a paper which has been forgotten, I think, in lots of places. It's about white light interferometry. White light interferometry is now very important. It's the basis of OCT. So the idea is that interferometry is very, very sensitive. You can measure very small phase changes, but it's not very good if you want to measure large phase changes. By using a broad band of different frequencies, wavelengths, then you can get around that problem and end up being able to extend the range that you can measure phases over. So that was in 1950. Back to the flying spot microscope, Young and Roberts published this paper. It was actually published in Nature in 1951. The flying spot microscope. They don't mention anything about R.C. Webb that I mentioned earlier. This is 2 years later. I don't know whether they knew about it. I actually knew Frank Roberts quite well. He was getting quite old when I knew it, but he was living in London when I was in Oxford. Now I'm back to the microphotometry again. Another Japanese person, Naora. Now apparently Naora was a student of Koana, and he carried on with that work. He published this one paper, which was actually in Science. So I don't know why this paper hasn't been cited more, really, but I don't think people on the whole know about it. It's basically a confocal system. In this case, it's a transmission system, but again, it's got this effectively a confocal pinhole. He calls it an iris diaphragm in front of the detector here. When I first saw this diagram, I thought, oh, this is interesting. It looks as though it's an interference microscope, but it's not actually. This path here is just for calibration. You don't have both the beams at the same time. You've got a mirror here that allow you to switch from the one beam to another in order to do the calibration. So 1951. Now I also knew Naora when I was living in Sydney before I went to Singapore. There was a meeting of the Australian Microscopy Society, and they presented a medal to Naora, and it was in Canberra, this meeting, I remember, and they asked me to present it to him, so that was rather nice. He spent he lived in Canberra. He was at Australian National University for much of his life, actually. Right, now we're getting on to what everyone knows already, which is Marvin Minsky. And, you know, he's often credited with inventing the confocal microscope. Well, I guess in some ways you could say he has. All the previous ones I've shown, apart from, well, they either had a slit, i.e. they didn't have a pinhole, or they didn't really produce an image. It was just a measuring device rather than an imaging device. So Minsky was the first probably to put all these things together and end up with a real confocal microscope. So you see, this is from his patent. He only published the patent. You can see a transmission system and a reflection system. And one of the things I wanted to point out here is that, you know, just to mention that this is a very long time ago, 1957, and you can tell that from the diagram. Look, this is his source of light. It's a lamp. You know, no laser. This was before the laser was invented, so he didn't have a laser. And even the photodetector, you see, is a very simple phototube here. Now, I think there probably were photomultiplier tubes by then, but he doesn't seem to show that in the diagram. The third thing that, of course, Marvin Minsky didn't have was a computer. So, you know, 1957, people didn't have computers attached to their microscopes in those days. So there are a couple of other things I wanted to say about this invention of Minsky's. Firstly, his name for it, he called it a double focusing optical instrument because it's effectively the light is focused twice. And the other thing is that he was obviously very well aware of the fact that you could get a resolution improvement using this, which is not really now the most important part of what a confocal microscope does. You know, the most important thing now is the optical sectioning, but he was well aware of the resolution improvement. Now I'm going to jump to structured illumination and a paper by Lukosh. Lukosh actually published a lot of really nice, interesting papers about the theory of super resolution. But this is one of his papers where he proposes effectively structured illumination. The idea is that you illuminate the sample with a pattern of fringes. So what that does is shifts the spatial frequencies of the sample so that they go through the imaging lens. That's a very clever idea that you can shift the spatial frequencies to get them through the lens. But then, of course, in order to get the final image, you have to reconstruct that. You have to move them back again to their real spatial frequencies. Nowadays, we do that with a computer. We take a few images, put it all in the computer, and then you do the reconstruction effectively in Fourier space, moving things around in Fourier space normally. But he didn't do it that way. Again, he didn't have a computer, so he couldn't do it that way. So his way of doing it was to do it optically, and basically using a second grating. So you see the system. You have the grating before the sample, and then another grating after the sample. So the grating diffracts the light to produce these different diffraction orders, and then the second grating combines these diffraction orders and puts them back into the correct spatial frequencies. He talks about in this paper, as well as doing straight structured illumination, he talks about making a near-field microscope like this, directly with the gratings. And then he also talks, he says the method may be used with coherent, partially coherent, or incoherent illumination. So that's what he was thinking of when he said incoherent illumination. I'm not really quite sure. He doesn't mention doing it with fluorescence, but maybe he had thought of doing that. Right, now we get on to the spinning disk microscope that was invented by Patran. So a couple of, they published a few papers around the same time. Their main reason for doing this was the optical sectioning property, but they again were aware of the resolution improvement, which they describe in one of these papers. Patran took out a patent on the idea of the spinning disk microscope. He called it the tandem scanning microscope. This works basically by having this spinning disk with lots of holes in. The holes act like the confocal pinholes. So it's like many parallel confocal microscopes. But more than that, it actually produces a confocal image that you can look at by looking down an eyepiece. So that was his idea. Unfortunately, Patran died earlier this year, actually. He was, I think, 96 or something like that. I met him a few years ago at the last World Microscopy conference in Prague. It wasn't that many years ago now. McCutcheon, we're going to go on to now. McCutcheon, he was a very interesting person. I never met him, but I did have correspondence with him on a number of occasions. So he had a paper where he talks about the super resolution effects of confocal microscope, and even doing confocal fluorescence, he describes. And also that you could make a near field microscope in the same way by scanning an aperture close to your sample. But he didn't seem to be aware of Minsky's patents. This was in 1967. I might add that he published another couple of very interesting papers about different subjects. I can't describe it now, really, but the interesting thing was that he was actually a medical doctor, so optics for him was just a sort of hobby that he played around with. So quite impressive, really. Right, now the specular microscope. So these are still widely used in the ophthalmology labs. The idea is that, again, it uses this idea of an off-axis arrangement. So the light comes in on one side of the lens and goes out on the other side of the lens. And in between, there's a spacer here that stops this light getting into that light. So you see that, again, it's got this arrangement of the illumination and the detection being separated. And this gives rise to a very interesting effect, which we call angular gating. So this actually gives a sort of it gives us a sort of optical sectioning effect, which you can combine with confocal sectioning. And it hasn't been used so much really in the normal confocal microscopes, but it's an interesting possibility that I think people might take this up again sometime in the future. The idea is that, of course, you see here, you see the light coming in and the light going out. You of course only produce an image if it's both illuminated and detected, right? So where these two rays cross is the bit you're going to image. So you get this optical sectioning effect that comes from this angular gating. Yeah, sorry, I've got this thing then. Yeah, well, let's carry on. Right, the scanning laser microscope. So this is, Davidovits and Egger published this. Rather interesting, you probably noticed that the Patran spinning disk microscope, Egger was involved with that, one of those papers. In fact, he was the first author. So he was at Yale, and Patran visited Yale from Czechoslovakia, so back in the late 1960s, so during the time of USSR. So at that time, Patran spent some time working in Yale. They developed this scanning laser microscope, and he cites Minsky as being the justification for building this sort of system. Yeah, here it goes. He thanks Patran for discussions leading to the formulation of the proposed microscope, and S. Baer for suggesting the application of a laser to Minsky's microscope. So Davidovits and Egger. Now we're going onto Slumber, now Slumber, he developed this system, which is a fluorescent system, and it's quite apparent that it's actually a confocal system, because you see that this system, you can see the detector is the source, here's the detector, and here's our pinhole. And he describes about how the pinhole basically means that you're only looking at the point you're illuminating. So he obviously understood about the idea of confocal imaging and applied it to fluorescence. This is a bit different one. This is Courtney Pratt. So he published this paper where you see the title, Microscope with Enhanced Depth of Field and 3D Capability. So the clever idea that they proposed in this was that you use a white light source and you focus the different colors at different depths. So that allows you to measure the depth within the sample by just separating the different colors. Quite an interesting concept, which has been applied in many other areas in microscopy since, actually, this basic concept. Now we're going on to, this is Sawatari, and Sawatari in 1973 published this optical heterodyne scanning microscope. And now this is an interesting thing. It's again, it's a confocal microscope. The illumination side, there's a lens on the illuminating side, which illuminates the sample with a small spot. But on the detection side, there isn't a pinhole. He uses a heterodyne system. So the idea is that instead of using a pinhole, you interfere the light with a reference beam. And this acts like it forms like a sort of synthetic pinhole, I like to think of it as. So you look at the correlation between the wavefronts of the light that comes from the sample and your reference beam. So it's a clever idea to another way of making a confocal microscope. And now we're back on Maurice again. Earlier I showed his specular microscope. This one is the scanning slit microscope that he developed. So you see in this one, again, it's you illuminate the light with a line of light, the object with a line of light. And then again, he's used this idea that Goldman had of writing the image onto a film. So the film passes through behind this slit and writes the confocal image line by line onto the film. Again, another example in ophthalmology. Right, and now we've got on to when I first went to Oxford. So I moved to Oxford in 1975, and we started. I moved there in 1974. We actually got this working microscope going within about 6 months of me getting there. It was a stage scanning microscope, very simple, really. You can see a helium neon laser. Actually, this one is a transmission microscope. You can see two objective lenses, the sample in between, and the sample is scanned mechanically using these voice coil actuators. And in transmission, of course, there's plenty of light, so there's just a photodiode to pick up the signal. This was the very first image we produced. You can see the image was displayed, no computer. So we scanned, it took a couple of seconds to scan an image. And so the image was displayed on a TV type display. Actually, I mentioned to the panel earlier that I did my PhD in electron microscopy. In the scanning electron microscope, this is exactly the way they used to do the display in the scanning electron microscope before the computer forms of it appeared. In fact, there was a student who was coupling up a computer with a scanning electron microscope when I was doing my PhD in Cambridge. So this was the very first image we produced. And we were actually also working on a beam scanning system. This is a picture of some blood cells taken with our beam scanning system and using galvo mirror scanning. But at the time, we mainly worked on this stage scanning because we were getting better optical images. Right. So this is a bit of history. This is me when I was younger, and some of our team. The work on the confocal microscope in Oxford was started by Rudy Kompfner. This is Rudy Kompfner, and Rudy Kompfner was a very famous scientist who invented lots of things, including the traveling wave tube was his most important thing. But he went on to become the director of one of the Bell Labs, the Bell Labs in Holmdel. And when he retired from Bell Labs, he decided that he was young enough to do two jobs rather than one. So he used to spend half his year in Oxford and half his year in Stanford. So that was how I got to know Stanford University quite well in those days back in the late 70s. This is Amar Chowdhury. He was my very first PhD student, or DPhil student, I should say. So he would have started in 1974. He finished in 1977. And together we published this paper, Image Formation in the Scanning Microscope. This actually, one of its claims to fame anyway, is I think it's the first paper to use the term confocal microscope. I haven't found an earlier one anyway. So he went on to become, he was from Assam in India, and went on to become the Vice Chancellor of the University of Guwahati. The other guy in this picture is Peter Hale. He was another DPhil student, but he wasn't working on the microscope at all. He was working on optical fibers, which was another completely different project, really, at the same time. The other one who was working on the microscope and who designed that scanning stage I showed you on the previous slide, unfortunately, he's not there because he's taking the photograph. So that was Julian Ganaway. And I'll mention him in a moment. Not quite yet, though, obviously. Yeah, this was, I'm not sure whether this was the first laser scanning microscope. It was a laser scanning microscope, not confocal, that was made commercially by Olympus in 1976. And early laser microscope. Yeah, so here we're getting on to Ganaway. These are pictures from Julian Ganaway's first paper on second harmonic generation imaging. So this was published in 1978. There was actually a conference proceedings that published it in 1977, but there weren't any pictures in the proceedings. And what this shows here, these two images, are basically showing two different depths from the same sample, demonstrating the optical sectioning effect. The idea is that any multiphoton type system, you get optical sectioning without needing a pinhole because of the multiphoton effect, the two photon effect in this case. Now, so I also had another paper in that same year, 1978, with Rudy Kompfner, where we pointed out various different types of scanning nonlinear microscopy that could be performed. And in particular, we mentioned the possibility of doing two photon fluorescence microscope and Raman scattering, coherent Raman scattering, i.e. CARS type microscope. And of course, it was much later that that was actually achieved experimentally by Denkin Webb in 1990, I think it was. These pictures of second harmonic generation were actually done using a CW laser. You know, we did actually do some experiments with mode lock lasers at that time, but only picosecond type lasers, because that's really all there were available at that time. So that was the work on nonlinear microscopy in those days. The other thing that I got very interested in when I was in Oxford was Bessel beams. So this was another thing that I guess I was a bit before my time, because Bessel beams didn't really take off until, again, around 1990, with the papers by Durnin and so on, where he calls them diffraction-free beams. But we were very well aware of what the Bessel beam would do. And we described in this paper that I had with Tony Wilson, this paper describes, as it says here, a wave with a zero-order Bessel function radial distribution, i.e. a Bessel beam, propagates without change. So we recognize this propagational invariance, the fact that it doesn't spread as it propagates. We also realized at the time that they did have some other problems. And if you put it through a microscope objective of a high numerical aperture, you don't get a single spot, you get a double spot. This is a polarization effect that comes about from using linearly polarized light and focusing the linearly polarized light. Nevertheless, I did take out a patent on confocal microscopy using a Bessel beam. That was taken out, this is the US one, 1980. I think the British patent was, I think, 1977 or thereabouts. So it's interesting. I never made any money from this. Various people have built confocal microscopes using Pascal beam since, but probably they don't even know that I had a patent on it. Right, confocal transmission. These were our first pictures of confocal transmission. So again, this is with Tony Wilson in Oxford, and 1979. And what we were showing here, comparing it confocal with a non-confocal, and the fact that we saw a sort of phase contrast effect, which comes about from the optical thickness of the sample. So that's quite an interesting effect, which has been used in some commercial instruments, actually, for looking at looking at blood cells and things like that. Also in 1979, Fred Brackenhoff, he was working on confocal transmission at the same time, and he published a paper. You see, this was in Journal of Microscopy, where he demonstrated the image improvement, the resolution improvement that you get in confocal transmission. So also 1979. Brackenhoff had another paper in that same issue of Journal of Microscopy, where he worked on an interference, confocal interference system. So it's a transmission system. So here's the diagram showing the light going through the sample and a reference beam and these interfering. And so you could measure the phase change through the sample. That was the idea of that system. Right, so through a lot of the 1980s, we did quite a lot of work on confocal reflectance microscopy. These are some examples of the time I left Oxford to go to Australia in 1989. So all these pictures were taken during that period in the 1980s, up until 1989. So you can see various nice things here. This is a stereo pair of pollen grain, confocal reflection. This is a color confocal reflection image using 3 lasers. So we had a red, a green, and a blue laser, each with their own detector and pinhole. And so this is the true color. And this is a picture of a leaf where we're looking down into beneath the surface of the leaf. So you can use the confocal sectioning to actually look inside the leaf quite nicely. And this is another one I think is an interesting one. This is a cell dividing. We're looking at the microtubules that have been labeled with 15 nanometer gold particles. And you will find many papers that say that you can't look at gold particles as small as that with a microscope. Well, you can. If you look at them with a confocal reflection microscope, you can easily see 5 nanometer gold nanoparticles. I'm not really quite sure why this technique hasn't been more widely used, actually. Right now, Fuji. I didn't mention it in what I wrote about my background, but I did actually also spend a year in Japan, and I was working in the lab of this guy Fuji, who published this nice paper. Again, it's this optical heterodyne microscope, but his reason for doing this was he wanted to avoid the lens. Or at least he almost avoided lenses. So the idea, it wasn't a confocal microscope, but he used this heterodyne principle to make a sort of synthetic lens. So effectively you use a reference beam, and again, by a correlation effect, you just measure the phase front of the object beam, which matches the curvature of the reference beam. And his idea was that he would then be able to use that with wavelengths that would belong beyond the visible spectrum. Yeah, and Kirster, so now we're getting a lot of different principles that we've already mentioned. Again, this is the off-axis type effect. You see the light comes down this side, up the other side, so we get this angular gating again. This is a confocal slit system, and again, it's for looking at the eye. So this is looking at the cornea of the eye, and he uses this clever idea of using here a mirror with well, it's actually a square with mirrors on the different sides. He had another one, I think, that had a triangular arrangement, triangular prism with mirrors on the sides of the prism. So interesting. Again, he mentions the optical sectioning characteristics. Yeah, so this is another of our old papers. This was one of our first confocal reflection images that was published, but not quite. I think there was one or two in the year before that. But this one, I think, was quite an important paper at the time because it demonstrated quite nicely the optical sectioning effect. So this is a conventional and a confocal image of the same sample that's taken in the same microscope. All you have to do, this is with the pinhole, this is if you take the pinhole away, then it becomes the same as a conventional microscope. And you see the images look completely different. Here, we're just seeing this stripe. And the explanation of this is that this sample is it's a silicon wafer that was tilted slightly. And so you only see the parts that are in focus. This bit and this bit are a bit above focus and below focus, or maybe the other way around. So here, you see, this one here is out of focus, but you don't see it. Here, you don't really even see that it's particularly blurred. So what this really demonstrates is the fact that the optical sectioning effect is actually a stronger effect than the defocus effect, which was quite an important revelation to us, I think, at the time. Another off-axis system, a really rather different one. This is for looking at measuring surfaces. So the idea is if you illuminate at an angle and then look at the light that's reflected from the surface, because of this off-axis arrangement, you can actually measure the surface height variations. This is another system. This is a commercial system, the deep field photographic microscope that was made by Chem Equipment Company in Illinois. And you see it's illuminated from the side here and detected up this way, and there's a camera to take pictures. So this is an early version of a light sheet microscope in the mid-80s. Around that time also was another commercial system made in the US by Irvin Optical Corporation. And I can't find anything about this company, but I did find this article in this magazine called Discover, which describes this. And this is showing a pair of images then of an insect. So this is a conventional image, and this is with this light sheet type imaging arrangement. Actually, this one looks very similar to pictures that we took of insects using confocal reflection microscopy when I was in Oxford. 1983, I had another student, Ingemar Cox, and so I'm going to show a couple of things from his work. The first thing he did was to couple up a computer to our confocal microscope. So this is from a paper which describes that system in 1983. Now, one of the things I wanted to point out here, 1983, I guess people lose track of time. computers were not quite the same then as they were now. In this system, our computer, we could store 3 images, 512 by 512 pixels. That was our total image storage. And this system, it cost us around £20,000, which was more than double the price of our first house. So you compare that with nowadays, how much it would cost to store 3 512 by 512 images. You could do it for $1. But anyway, almost. So you see how the computers have changed over the years. This is an example of using computer processing on this was actually done in my early years when I arrived in Sydney. So again, this is a silicon wafer and silicon chip. And this is an autofocus image. This is a surface profile image. If you just store those 2 images, you can reconstruct this sort of isometric view of what that surface looks like. So it produces really rather nice images like that.
Colin, can I just remind you, sorry, we only have about 10 minutes now.
Ah, right, okay.
Don't rush through, but be selective of the slides which matter to you most.
Yeah.
And we don't need to do the quiz if you rather have the time. So I'm willing to give you the 10 minutes.
I'm quite close to the end.
Just make sure.
It's best if I just carry on.
Sure, yeah, that's fine. Okay? Just so you know, because we get.
Okay. Yeah, I wasn't looking at the time.
No, that's fine. Of course, it's super interesting. So just to remind you.
Okay, so confocal interference, reflection interference microscope we built when I was in Oxford with Doug Hamilton. This is a picture of our system. A lot of this gear, by the way, is still in the lab of Martin Booth in the engineering department in Oxford. I think it's hidden under some in a cupboard or something. But anyway, we built this confocal interference system in those days, 1982. Now, Ingemar Cox, he published these papers, confocal fluorescence images, in 1984. And I don't know why, but this paper has been not very well cited. But it's really predates pretty well all the other confocal fluorescence papers that I know of. Another whole area we were working on back in those days was phase imaging. And the method we were mainly working on was using basically what we call now differential phase contrast. So this uses two D-shaped detectors, and you subtract them and you end up with a phase image. And so these are some images of cheek cell taken by different geometries of the detector. And this is showing here, it's very, very sensitive. This is actually a single monolayer on a glass slide here that you can see quite clearly. Right, and this is the last slide. And I just wanted to say something about the early history of the commercialization of the confocal microscope that people might not know about. We started, when I was in Oxford, we started a company called Optoelectronics. It was formed in 1980. We launched a commercial system in 1982. We sold a few of them. But eventually the rights to that design were bought up by LaserSharp, a division of IBT Durabilia. So they re-engineered it to produce this system here, which they called the SOM 100. That was launched in 1984. And then subsequently, this company was bought up by Biorad. And then they acquired the rights to this in 1987. But as we all know, they developed the Biorad MRC-500 using a design from Cambridge University, Brad Amos's design. and they just got rid of our design. So that was the end of our microscope. So I think that's the last slide. So there we are. Yeah. So thank you. I've got through it just about in time.
Yes, thank you very much. I just thought I remind you. So thank you very much.
Yeah. Okay.
I think it was if you could stop sharing, actually, that would be nice.
Yeah, that's
so
good.
We can also see you. Thank you very much. So I think it's also, maybe in the interest of time, if people want to ask questions, because rather than do the quiz, because it's a bit disruptive. So I see that Michael Gilligan has actually helped in providing PDFs in the chat. And also what is actually quite lovely, he shared a book and some information about the company, the Irvine Optical Corporation, with a nice young lady who's actually next to the microscope if you all click on the chat. So I think this is quite fun. So I'm very happy to have heard the history of the confocal as you have told it, and because the origin of Imaging ONEWORLD is actually in Cambridge. We have obviously the history of the biorad microscopes. We have heard many times from Brad Amos and John White, but actually seeing all the developments of confocals, it reminds me also light sheet microscopy in the last 10 years. I mean, everybody is puzzled by how many permutations and how many different variations there are, but I guess, I mean, I can see and I know the confocal microscopy kind of field was just the same. I mean, so many people made so many different permutations, and some of them became commercially viable and successful, but others maybe not so, but not, you know, they're just all these different ways of imaging. So this was wonderful to see. Would anyone see Ian Doby is actually in the audience, and so is actually Jonas Ries. If you may actually, I don't know, anyone want to ask questions directly would actually be nice, or just say hello. Or maybe people need to go because there's Ian Doby. He actually put the question in. I would just ask it yourself.
I don't know if I put a question but a comment. The original Logie Baird TV system was a Nipkow disc rather than a flying spot.
Yes, it was actually. You're quite right. You're quite right.
That was an absolutely fascinating talk. Thank you very much. And I've heard of some of these, maybe 10% of the papers which you mentioned in the last hour. It's been, I mean, a real tour de force of things which we should remember and don't. So thank you very much. That was very, very entertaining and interesting.
Yeah, thanks for pointing out. It was, of course, the Nipkow disc. I knew that.
All us old farts got to remember.
I think it was only recently that I think that people started to work on, or like being able to commercialize systems which were no longer based on the Nipkow disc, isn't it? So there are kind of variations now. Anyone else would like to ask questions? I think there's Nick. I don't know who's developing systems, but I mean, I just think it was really wonderful to have all these technologies. I mean, we did prepare the quiz, and one of the questions was obviously for the different systems. So there was the microscope which was used for eye and cornea imaging. I was looking at this. Sorry, I kind of the specular microscope, and I was just thinking, would that actually be useful for other tissues?
Yeah,
so I'm just hijacking the quiz.
We had a paper. I had a paper about that sort of technique a few years ago, where the images we actually published were of ball bearings, and we were using this as a way of distinguishing. You know, you can, if you do a confocal image of a ball bearing, you can focus onto the surface, but you can also focus onto the center of the sphere. And this is actually a cause of errors that you get when you're doing surface profiling using confocal. So it's quite interesting that it's got a very important effect actually in a useful area.
Because you also showed the ultra microscope, and one of the questions was, what was the ultra microscope used for? And I don't know if people noticed it or in the audience, but the answer was imaging small scattering particles. So I guess, I mean, it also is a reminder how many different components needed to come together, because I guess it was used for scattering particles because the camera sensitivity or detection sensitivity was maybe not as high, or fluorescence wasn't really used that much either.
Yeah. Well, fluorescence really, I think, was later, 1911. I think the first papers on, because it required, in those days, it required using UV light in order to get the luminescence.
Yeah. Okay. And because also, I mean, I remember fluorescent microscopy being quite a special thing to do in the lab, and like 30 years ago, and then going from fluorescence microscopy to confocal fluorescence microscopy, it was such a big step in terms of sensitivity, and also what you were saying about computers. So the invention of computers, I mean, it's being able to store 3 images. That's initially, for my PhD, I only had floppy disks where I kind of saved 3 images on a floppy disk. So from the confocal, and we underestimate actually that wasn't actually available. To see the images you've shown actually from all the different specimens you've taken, I mean, that's spectacular, really amazing.
Yeah, so the other thing, we couldn't actually store, when I say storing 3 images, I'm talking about XY images, not stacks. So, you know, to do any sort of 3D stuff was, you had to juggle around with lines of images at a time in order to make available, make use of the space you had. So
yeah, I remember. It was 512 by 512 pixels, and that was already amazing. And also, you had one question. This, I'm just taking the quiz out and doing a bit faster, about Guana and Naora's confocal system. And the question was, what is it used for? And it was microphotometry. But I actually made up some extra answers, and I was surprised because it was actually spectrometry, but it was used for microspectrometry, I think you said in there, so in your talk. So I mean, it just shows how many versatile imaging techniques people used, and how many different angles they came from. So.
Well, a lot of the application of the microphotometry was basically reading films. You know, you write data onto films, and then you've got to read it in some way.
And Ian is saying, like, nowadays we're storing, he's making us kind of laugh, 200 gigabyte stitched 3 color set stacks, and we do that probably within minutes, you know? And also a reminder, I mean, if you want to join us next week at the same time, actually Catherine Lau is going to speak about in-chamber fluorescence microscopy imaging to guide cryo-electron tomography sample preparation. So you might, I mean, because we're talking about the combination of EM and light microscopy and confocal and imaging across so many different variations. So if you're interested, and everybody else, obviously you're most welcome to join us next week again. So thank you. I think several people are thanking you. And I mean, I'm sure, I mean, they're also kind of really, if anyone still wants to ask a question, but otherwise, unfortunately, our time is up today, and it's always the hardest to actually kind of leave now the room, because we can't go for coffee or drink. You can only go to sleep now, and we can go.
Oh, I think I might have to. I think
have
I
a
might
tea and cake.